It's a pleasure to be here today. My name is Ryan Urbanowicz, and today I'm going to be talking about some new work of ours on developing a new piece of software that is an automated machine learning analysis pipeline for biomedical data mining. But first off, I want to get everyone on the same page and just briefly refresh our minds as what is machine learning, or ML. And this is a definition from Samuel Arthur that I like. It defines machine learning as a subset of artificial intelligence in the field of computer science that uses statistical techniques to give computers the ability to learn or progressively improve performance with data without being explicitly programmed ahead of time. And this is really key to machine learning, is to learn from data on its own. And the big picture goal here is to learn useful generalizations. In other words, we want to find patterns or associations that we can use to make predictions. Machine learning is not typically designed to demonstrate causality, but again is mostly looking for these associations. And our typical pipeline would look like, we'd start with data, we have some machine learning algorithm that we apply that trains a model, and then we can take this model and apply it to make predictions. When we're dealing with biomedical data analysis, there are lots of really common challenges. For instance, the scale of the data, how many features, how many instances, how noisy is the signal in the data? Is there missing values across our data set that we have to deal with? If we're doing classification, are our class ratios imbalanced or balanced? Another big one is the complexity of the associations between our predictive variables, as we see here illustrated on the left. In this case, we have genetic, environmental, or epigenetic, and the phenotypes or outcomes we're interested over here on the left. And so these can be very simple, like Mendelian associations. We can have complex interactions, also known as epistasis. We can also have heterogeneous relationships where different features contribute to the same or similar phenotype, or we can have some messy combination of a lot of these. Some other challenges include, dealing with covariates, which is not something that's always thought about in machine learning. How do we deal with rare variation in variables in our data? And of course, in medicine and a lot of fields, interpretability is really key. And we can start to address the interpretability of our machine learners by looking and asking questions about what features were important in our data set, what variables, to characterize these different complex patterns of association and distinguish them from simple ones that exist in our data, and to use them to make individual predictions on new data. So just stepping back, artificial intelligence has become really popular in biomedical research of late. And so I just want to ask the question, what's the role of AI in biomedical informatics? Here are just some thoughts. But basically, there are effectively an infinite number of machine learning pathways or pipelines that we could design to analyze data. And there's all these questions, including how should the data be cleaned or transformed? What features could be or should be engineered to make machine learning more effective? What features from our data set should we select and keep rather than ignore to make our machine learners operate better? What model or machine learning method or algorithm should we use? Which one do we expect to give us better performance? And what parameters for running those methods will yield optimal performance? And these answers may differ as we go from one problem to the next. AutoML has become a hot topic of late. And basically the idea is to simplify this whole process for new users, or to optimize our ability to apply machine learners to new problems as we go from one to the next. So here we have a really simple pipeline example where we start with raw data, usually requires some cleaning. Then we need to do some sort of feature processing, followed by the modeling itself. We want to pick a classifier, in this case a machine learning algorithm, and we might want to optimize the run parameters for that method to get it to give us the best models possible. But this whole process can be tedious, and it requires a lot of expertise to be done correctly. And so there are questions in AutoML about how do we design automated machine learning methods that can automate parts of this pipeline. So in this case, we're just automating the selection of an algorithm and the optimization of its run parameters to get it to work better and better to find stronger signals in the data. Or we can expand this to try and automate larger parts of the process. And in fact, that's something we're trying to deal with in this talk. There are lots of different open source auto machine learning tools out there currently, and there's new ones coming online every year, every month even. Here's just a small array of them. But one thing that they have in common is that generally they're trying to optimize finding one best model that was trained on the data and then go on from there. So the motivation of the current work, we've developed this new method, or this new pipeline called AutoML-Pipe BC. And I just want to review some of the goals of this new piece of software that is now currently available for anyone to download and use. So basically, we wanted to automate and parallelize phases of machine learning analysis pipelines. In this case, we're just focused on binary classification tasks, because as you go deal with different kinds of outcomes, you'd have to change the pipeline a lot. So we wanted to focus, at least initially, on binary classification. And basically, we wanted to obtain a rigorous and thorough look at performance. We also wanted the capability to handle big data, right? So to handle data sets where there are lots of features, it could be genomic data or lots of instances. And so in our pipeline, feature selection, as you'll see briefly, plays a really big role in the ability of this pipeline to automatically handle this issue. We also wanted this pipeline to have the capability to deal with typical data challenges, such as data missingness, imbalanced data, and dealing with data normalization that some machine learning methods require for both efficacy and interpretability of the models they produce. And then further, we wanted this pipeline to be able to take complex associations into account, in particular epistatic interactions, which I alluded to earlier, that are not easy for all methods to detect or account for, especially early stages of feature selection, where it's really important. We don't want to remove potentially informative features before we get to modeling itself. Some just broader goals of this pipeline is to provide a standard of comparison for other AutoML tools. There's this lingering question out there: is does AutoML actually do better than an educated data scientist sitting down and creating their own machine learning pipeline from scratch? We also wanted to have a pipeline that could compare different machine learning modeling algorithms that were developed in the future to one another. So this provides a framework to do that. And also to compare performance between target data sets. So if you have different data sets with different subsets of features in your data that you want to compare, or you want to run your data set with or without covariates to evaluate how well machine learning could capture the signal in the presence of covariates or not, you could also use this pipeline to tackle that question. Another issue is we want our models to be interpretable. And so this pipeline facilitates your ability to interpret feature importance within and across models and machine learning algorithms, as well as to try and characterize these complex patterns in the data. And lastly, to apply models to future data. Once we've trained these models, we've picked our best ones, we want to easily be able to apply them to make predictions in the future, either in the clinic or elsewhere. So this is a schematic of the pipeline as a whole, and I'm going to talk about bits of it as we go through this talk. But at a high level, this pipeline, we have our raw data coming in at the top. We have in pink a pre-processing and feature transformation phase of the pipeline. Then down here, it goes to the right and then connects here to the left. Then we have a feature selection phase, which is second. And then we have machine learning modeling itself. This is the part that most people are familiar with or that they've dealt with in terms of trying to tackle machine learning modeling. And then lastly, we have post-analysis. So I'll get into this a little bit more as we go. But this is everything that this pipeline automates on its own. And we've taken this pipeline, and at least in this talk, I want to talk about our application of it to a data analysis of obstructive sleep apnea, or OSA. This data set came from the Sleep Apnea Global Interdisciplinary Consortium, or SAGIC. It included over 3,000 de-identified subjects who underwent polysomnography, or PSG. And our target outcome here was the Apnea Hypoxia Index, or AHI, which measures events per hour. And basically looked at two different cutoffs, a liberal and conservative one of either AHI greater than 5 or greater than 15, just to see which one would be more predictive. And furthermore, we compared data sets with different groups of features being added. We had 5 categories of features, including demographics, comorbidities, symptoms, craniofacial photo features that were derived, and intraoral photo features that were derived. In total, we had 85 features. And in this study, we analyzed just demographics alone, and then demographics plus comorbidities, and so on and so forth, progressively adding these more sophisticated features to the data set to ask the question whether they allowed us to predict obstructive sleep apnea better or worse. So across this analysis, these were all of the runs that we completed using our automated pipeline. We used these 2 different AHI DBHi cutoffs, and these different combinations of features that I mentioned just a moment ago. Getting back to the pipeline, the first phase is of the preprocessing is the exploratory analysis and basic data cleaning. This does, very basic components of the machine learning process, but what's really nice is it also outputs all of the summarial information and figures that you might want for this initial phase. So here we see a bar plot of our class imbalance. In this case, we're looking at an AHI greater than 5 and this particular combination of features. Here we have a feature correlation plot so that we can go back and ask what features were correlated in our original data set. We can look at the counts for instances, features, and the pipeline automatically separates categorical from quantitative features based on a user-defined cutoff. But also the user can specify what features they want to be treated as categorical or quantitative manually. This exploratory analysis also outputs appropriate univariate statistical association analysis, as well as either box plots or bar plots based on whether a given variable is categorical or quantitative. So this exploratory analysis gives us an initial look at the entire original data set. The next phase in pink here, we have we apply cross-validation, and this is important so that we have a less biased look at our data set. And we do this prior to any other component of the pipeline. So notably, everything that happens beyond this phase is happening within one of K training or testing sets. So by default, in this study, we've looked at 10-fold cross-validation, so K here is 10. This phase also includes transformation of features. The pipeline automatically applies a standard scaler, and this is important for machine learning methods like logistic regression or artificial neural networks to get the variables within a standard scale. Next, we apply data imputation if there's any missing data in the data set. After that, we have feature importance and selection. And I'll just mention briefly that this pipeline implements two different filter-based feature importance methods. One focuses on univariate associations, that's mutual information, and another, multi-surf, is able to conduct an efficient look at feature importance while taking feature interactions into account. So if you are interested in complex epistatic interactions in your data set, this is a really important thing to include in feature selection. And we actually evaluate both, and then we combine the results using something called collective feature selection, which looks at the union of features identified as being relevant by either one of these univariate approaches. So basically what happens is, if either method finds evidence that a feature is valuable, it's going to keep it and pass it on for machine learning modeling. It's a pretty liberal feature selection approach, but the user can actually set most of the run parameters of this pipeline to customize it further to the needs of their given study. So just real quick, looking at some of the output from this feature selection component of the analysis, we have for one of our analyses, we have the mutual information scores ranked, as well as the multi-surf scores ranked. And so you get these visualizations automatically from the pipeline, which is another nice bonus. Moving on to the heart of the pipeline is the machine learning modeling. So here we've implemented 13 different machine learning algorithms, including many that I'm sure some of you have heard of, like logistic regression, naive Bayes, random forest, and some more sophisticated methods like XGBoost, artificial neural networks, and so on. And we've actually also implemented 3 rule-based machine learning methods that our lab happens to be specializing in research-wise. And basically, we've implemented them here to ask further questions about how their performance compares to other state-of-the-art methods out there in the field. And for every one of these methods, the pipeline automatically optimizes hyperparameter performance, and then we train a final model of each within each cross-validation set. And ultimately, we evaluate the performance of all methods across all classifier metrics and consolidate the results for the user to look at. And so down here, we have the interpretation phase, is where it brings all those results together, creates plots, and so on and so forth that I'll show you here in a second. Okay, so here is part of the evaluation metric output. We can see all of our machine learners here on the left, and we see all these classification metrics that you might normally care about here at the top. And so this is just a summary of the results, so you can see an example of what the pipeline outputs, basically giving the user pretty much anything they would need to evaluate performance. Here's for modeling. It also automatically generates ROC and PRC, precision recall curve plots. Again, comparing machine learners. Here, we're looking at the average performance across all tenfold cross-validation runs for each algorithm, allowing us a nice way to compare machine learning performance in our analyses. And so in this particular case, because we have a class imbalance, we actually found in this particular analysis that XGB, or extreme gradient boosting, performed the best. And so we're going to focus on that a little bit further, and you'll see that LGB actually performed best later as well. Another thing the pipeline outputs is for each machine learner, in this case XGB, we're going to pick on that for a moment, we have that same average performance that we saw in the last plot compared to other machine learners, but we can now see its individual cross-validation runs and the variation between those runs. We can see that for precision recall curve. We also output bar plots showing all the machine learner performance comparisons for every evaluation metric. Here we see our machine learners on the x-axis, and in this case, precision recall curve, or the area under the curve for precision recall curves here on the y-axis. And here in this plot, we see balanced accuracy. You can go through and look at all of these results at your leisure. Another thing the pipeline outputs is feature importance estimates for every machine learner. Feature importance can be estimated internally by some machine learning algorithms, but we actually universally also apply the option for users to apply the same type of permutation feature importance estimation so that you're evaluating feature importance the same way for all methods. And we can look at these bar plots for any given algorithm that was run in the pipeline. So in this case, just an example, BMI, not surprisingly for obstructive sleep apnea, comes up as the most important feature in this particular analysis. Another nice thing that we've added that's brand new to this pipeline that we think would be really useful for interpretation of machine learning results downstream is what we're calling a composite feature importance plot. So what you're looking at here are the relative feature importance estimates for every machine learner stacked on top of each other for each variable, and then we rank them. So we can look at consensus of feature importance across our machine learning algorithms, but we can also look at the individual colors. So red here is artificial neural network, and we can see how each algorithm viewed each feature in terms of its importance to that particular model as well. The pipeline also does nonparametric statistical comparisons. I won't get into the details here, but basically every kind of comparison you might want to examine, whether different machine learners are performing significantly better or worse than each other, or if different data sets you're analyzing and comparing to each other had better or worse performance. Another nice feature of the pipeline is it automatically outputs a PDF report with all the primary statistics and features, exploratory analysis images, ROC curves, etc., all in one nice little PDF packet. Obviously, you can't see the details here, but it's just meant to illustrate that it gives you this automated report as a starting point from which you can dig into all of the results that are saved. And then lastly, the algorithm also is set up to be applied to new data in the future, to data that you might want to apply to new patients, or maybe you have some replication study at another site, and you want to see how the machine learning algorithm performs in that secondary group of subjects. And so what happens here is the pipeline automatically saves all machine learning models and everything you'd need to boot them back up and reapply them. And there's a part of the pipeline allows users to easily load their data and rerun it on these new data sets so they can evaluate the models further. Just a quick note on implementation. You can find this whole machine learning pipeline here at this link. It includes a detailed README anyone with very basic Python experience would be able to apply this algorithm on their own very easily. It's designed to be run either locally within a Jupyter notebook. It can also be run locally from a command line. And we've also set it up to run in parallel because when you're running a lot of data sets and a lot of machine learners, it can be computationally expensive. So it's set up to run on our UPenn computing cluster, but you could also pretty easily apply it for cloud computing as well. And there's also some other notebooks that allows users to take the output of the pipeline and do a lot of extra things like regenerate figures with new labels, with new subsets of features, create decision tree illustrations, and so on and so forth. And here we see the individual phases of the pipeline that you'll find in this repository on GitHub. Quickly back to our sleep apnea study, I'll just mention the results here. I mentioned that we were comparing progressively adding features. Up here at the top, we see an AHI cutoff of greater than 5, and down here, greater than 15. You can see greater than 5 performed a lot better for precision, area under the curve of the precision recall curve. And what we also observe here is that as we went from demographic variables to comorbidities, there was really no improvement. But when we added symptoms, we did see a pretty significant jump, and a small jump when we further added craniofacial photos. These results were showing the results for all machine learning algorithms, I'll note. When we focused on the best performing machine learning algorithm in this run, like light gradient boosting, and then we actually conducted a follow-up significance analysis, we found that the addition, again, of the comorbidities didn't have a significant improvement, as you can see here. But as we went from either of these and added the symptoms variables, we did see a significant jump. But ultimately, this change in performance going from that to adding craniofacial photos was minor and didn't actually meet the significance criteria. So in summary, we have this new pipeline that facilitates application of established and novel machine learning algorithms as part of an automated, rigorous pipeline. And in our OSA application, we found in general that AHI greater than 5 gave us more predictive models than AHI greater than 15. LGB, or light gradient boosting, in this case was the best performing machine learning algorithm. Looking at LGB models and precision recall area under the curve as our target metric, we found that the symptom features significantly improved model performance, and craniofacial photos improved it slightly, but not significantly. Future directions, we want to extend this pipeline to also tackle multi-class problems and quantitative outcomes. We also are in the works of creating a deep learning version of this pipeline. We want to be able to use this to compare other automated machine learning algorithms. So use this as a standard of comparison to start to ask the question, does AutoML in the way it's been presented so far actually do better than just running a variety of machine learning methods like this and comparing their performance? And then we also want to use this to evaluate novel machine learning algorithms like the ones our lab develops and apply it to a variety of new biomedical data in the future. And with that, I want to thank all my collaborators, especially those in the Sleep Center here at UPenn, including Alan Pack, Brendan Keenan, Ulysses Mengelen, and many others, a number of great students, including Robert Zhang. And you can find more information about our lab projects on this GitHub repository, as well as some informative discussions about our research here on YouTube. And of course, I want to acknowledge our funding, and thank you again for the invitation to get to come and speak to you all today. And with that, I'll take any questions. Thanks again.